But the IC50 is literally 40 nanomolar in the primary screen. That is not noise, that is a signal. That compound is doing something real.
40 nanomolar? That's so tiny. That means it's incredibly potent, right? That's the drug. We found it. We found the drug.
I'm already calling it compound K7, trademarked. We file the patent Tuesday. The compound library is our competitive moat, and I will not apologize for that framing.
The compound library is absolutely the moat. I've been building mine for 3 years, in my shed, officially for research purposes.
It is mildly suspicious that a compound produces 40 nanomolar inhibition in a primary screen at a single concentration, with no dose-response curve, no counter screen, and no orthogonal assay.
What color was it?
Stop! We are going to cover this properly and correctly. Today we are covering how drugs are actually found. Not by accident, not by luck. Found deliberately through a systematic process. Hit identification, hit to lead transition, and lead optimization guided by structure-activity relationships. This is the engine that runs pharmaceutical drug discovery, and several people in this room have significant misunderstandings about how it works.
I understand it. I just don't respect the timeline.
I'm Dr. Seeley. With me are Bobby, Kelpie, Crabzilla, Squiven, and Patar. Let's begin.
Hello to everyone at home. I want you to know I am already very emotionally invested in compound K7.
Subscribe to this podcast. You are sitting on an untapped information asymmetry. Every episode you miss is a competitive disadvantage. Someone out there is listening right now who will use this knowledge before you do. Subscribe.
Before you can identify a hit, you need a target. A drug target is typically a protein, an enzyme, a receptor, a transporter, or an ion channel, whose activity is causally associated with a disease state. The hypothesis is, if I modulate this protein's function, I can change the disease outcome.
Modulate meaning mess with?
Inhibit, activate, block, or stabilize. Mess with is functionally accurate, yes.
And most hit-finding efforts are looking for inhibitors. An inhibitor sits in or near the active site of the protein and prevents it from doing its job. You're jamming the machine.
That is competitive inhibition. There is also allosteric inhibition, where you bind elsewhere on the protein and induce a conformational change that disrupts activity, and positive modulators, which enhance function. The biology of your disease determines which mode you're after.
I want to flag that target choice is also an IP strategy. You want a target validated enough that investors believe the biology, but novel enough that you have freedom to operate. This is a real conversation I have with portfolio companies.
It is mildly revealing that the scientific and commercial criteria for target selection are occasionally in direct conflict with each other.
Once you have your target, the traditional approach to finding molecules that interact with it is high-throughput screening, HTS. You take a compound library, sometimes 100,000 molecules, sometimes 1 million, and you test each one for activity against your target protein. Automated liquid handling, microplate readers, thousands of data points per day.
1 million random molecules? You just try all of them?
It is molecular speed dating at industrial scale. You swipe right on everything and wait to see who calls back.
The hit rate in a typical HTS campaign is between 0.01 and 1%. So from 100,000 compounds, you may identify between 10 and 1,000 that show inhibition above your threshold, usually 50% inhibition at a fixed concentration.
So most of them do nothing?
The vast majority. This is expected. The chemical space of all possible drug-like molecules is estimated at around 10 to the power of 60 structures. Your library of 1 million compounds is a rounding error.
But our library is still a proprietary rounding error, and I intend to monetize it. I'm calling it HTS as a service. We own the hits. We license the validated leads. We're the picks and shovels play in drug discovery.
It is mildly concerning that this business model already exists and is called a contract research organization.
Here is where it gets genuinely depressing. Your 0.5% hit rate isn't all real hits. A large fraction of what lights up your screen is chemical garbage that has nothing to do with your target. It just looks like a hit.
These are called PAINs, pan-assay interference compounds.
Pains?
Molecules that appear to hit virtually every biological target they are screened against. Not because they are broadly active, because they are chemically reactive frauds that deceive your assay.
How?
Several mechanisms. Some PAINs compounds are fluorescent, They absorb or emit light at wavelengths that contaminate your fluorescence-based readout, making it appear as if they are inhibiting signal when they are simply polluting the measurement. Some are covalently reactive. They modify your protein or your detection reagents non-specifically. And some form aggregates, microscopic particles in solution that adsorb your protein, sequester it, and make it appear inhibited because it is no longer free in solution.
The aggregation mechanism is my personal favorite example of chemical deception. The compound is not binding to the protein. It is kidnapping the protein and hiding it. Your assay reads inhibition. What actually happened is abduction.
Like when you lose something because you cleaned up.
That is a surprisingly reasonable analogy for non-specific sequestration.
Bale and Holloway published a landmark paper in 2010 identifying structural alerts, specific functional groups, and scaffold associated with pain's behavior. The tragedy is that by the time it was published, thousands of academic papers had reported pain's compounds as promising drug hits. Entire research programs had been built on them.
It is mildly tragic that a significant fraction of published drug discovery literature from the 2000s may constitute a library of chemically validated false positives.
I want to reframe this. If you build pains filtering into your screening cascade before anyone else, your hit quality is higher. Your validation rate is higher. Your probability of clinical success is higher. This is not tragedy. This is a sourcing advantage. I would fund the better filter.
Some estimates suggest that in certain disease areas, up to 25% of published hits from academic HTS campaigns are pain's compounds. The clinical translation rate from academic hit to approved drug is approximately 0.1%. These facts are not unrelated. Before we discuss hit validation, I need to raise something. Last week, Pitar recorded a full episode on the normal distribution. Specifically, why looking only at the mean or any single summary statistic is insufficient, and why you must look at the full shape of your data. She showed that two data sets with identical means, identical standard deviations, and identical ranges can have completely different underlying distributions. She covered bimodal distributions, skewed distributions, and what it means when your replicate IC50 measurements don't look Gaussian. It was directly relevant to everything we are about to discuss. I was there.
Oh, I have been meaning to watch that.
You were supposed to be there in person, Bobby.
Oh,
None of you were there. Not one.
I was running an extraction in the shed. It was at a critical temperature.
I had a board call. The timing was non-negotiable.
I genuinely thought it was next week. It was in my calendar as next week.
Pitar spent 12 minutes explaining why a compound that shows a mean inhibition of 60% across 12 replicates might be doing something completely different depending on whether that data is normally distributed, bimodal, or has two outliers pulling the mean upward. Alone, to an empty room, and
It is mildly symbolic that the episode most directly relevant to why reported hit rates in the literature are misleading had an in-person audience of 1.
I didn't mind. Tomorrow I'm doing significance testing.
yes, you are, and I will be there. The rest of you will also be there.
I will be there. I am setting 4 alarms right now. I will be in that room.
Noted.
I'll check my calendar.
It is not a suggestion. What you need to know for today. Do not report a mean IC50 without knowing what your replicate distribution actually looks like. A bimodal IC50 distribution across your replicates is not a measurement error. It is a signal that something interesting or something broken is happening in your assay. Pitar will cover the statistical framework properly next week. Moving on. After your primary screen, you do not celebrate. You perform hit confirmation. Every compound that showed activity goes back into a dose-response experiment. You test it across a range of concentrations and fit the data to determine an IC50.
The IC50, the concentration that inhibits 50% of your target's activity. This is your potency number. Lower is better. Low nanomolar is excellent. Millimolar is basically water with ambition.
What's in the middle?
A micromolar IC50 is typical for a primary hit. It tells you the compound has some affinity but is not yet potent enough to be a drug candidate. A low nanomolar hit in a primary screen makes you suspicious. Either you have found something genuinely tight binding or you have a pain's compound behaving badly. Both need the same next step. After dose-response confirmation, you run orthogonal assays. You test the same compound for the same biological effect using a completely different detection method. If your primary screen was fluorescence-based, your orthogonal assay uses a different readout. Radioactivity, mass spectrometry, thermal shift assay, or an enzyme-linked detection system. The question is, does this compound produce the same effect through a method that pains cannot deceive?
And if your compound only lights up in one assay format, it is almost certainly lying to you. Real hits survive multiple lenses. Pains fail the moment you change the detection method.
Direct binding confirmation is also required. Surface plasmon resonance, isothermal titration calorimetry, or NMR. These techniques tell you the compound physically associates with the target protein with a measurable dissociation constant, KD. IC50 can be misleading. KD is harder to fake.
So you need multiple independent proofs that the thing actually works?
Dose-dependent activity in at least 2 assays with mechanistically distinct readouts, plus direct binding evidence. At that point, and only at that point, do you have a confirmed hit worth investing in.
It is mildly unsurprising that the compounds most difficult to confirm as genuine hits are precisely the ones that generate the most initial excitement.
It's like checking if your keys are actually in your pocket or if you just felt something.
That is a reasonable description of the difference between a primary screen result and a confirmed hit. Yes.
Confirming a hit is not the same as having a drug candidate. A confirmed hit then gets assessed on drug-like properties, criteria that predict whether a molecule can realistically become an orally bioavailable drug.
Lipinski's rule of 5. One of the most cited observations in medicinal chemistry, technically a guideline, frequently misunderstood as a law.
Described by Christopher Lipinski at Pfizer in 1997, derived from an analysis of the physicochemical properties of marketed oral drugs. 4 criteria, all coincidentally multiples of 5. Molecular weight of 500 Daltons or less. logP, a measure of lipophilicity, of 5 or less. No more than 5 hydrogen bond donors, meaning NH and OH groups, and no more than 10 hydrogen bond acceptors, meaning nitrogen and oxygen atoms.
Why does the weight of the molecule matter?
To be absorbed from the gut into the bloodstream, a molecule must cross lipid bilayer membranes. Very large molecules cannot do this efficiently by passive diffusion. Molecular weight under 500 keeps you in the diffusible range for most oral drug candidates.
And logP is the partition coefficient between octanol and water. It tells you how much the molecule prefers fat versus water. Too hydrophilic and you can't cross the membrane. Too lipophilic and you aggregate in plasma, saturate the liver, and get metabolized before you ever reach your target.
So there is an optimal greediness profile. Some lipophilicity is an asset. Too much is a liability that kills the deal. This is also how I think about leverage.
It is mildly accurate to describe optimal logP as greediness. That does not destabilize the business model.
The rule of 5 predicts that violating more than one of these criteria significantly reduces the probability of oral bioavailability. There are exceptions: natural product-derived drugs, antibiotics, macrocycles. But as a first-pass filter for small molecule programs, it remains extremely useful 40 years after publication.
What happens if your hit fails all 4?
You grieve, briefly. You look at whether intravenous formulation or inhaled delivery is appropriate, and then you go back to the library.
Or you out-license it to someone with a higher risk tolerance and focus on your next scaffold. Not every compound deserves your balance sheet.
This is where structure-activity relationships become the central tool. You have a confirmed hit with acceptable drug-like properties. Now you systematically modify the molecule and measure how each change affects activity. Every position on your scaffold, every functional group, every ring, you explore what happens when you replace, remove, or add something. This builds your SAR profile.
You just change little bits of the molecule and test what happens?
Systematically change specific positions with a hypothesis about why you are making each change, and then measure the outcome. You are building a map of the molecular recognition between your compound and your target, which regions are essential, which are tolerant of modification, which can be exploited to improve other properties.
And this is where medicinal chemistry becomes genuinely creative rather than just synthetic. You're not only chasing potency, you're simultaneously optimizing metabolic stability, selectivity, solubility, permeability, and improving one property often breaks another.
Multivariable optimization under pharmacological and commercial constraints. Someone should be applying generative machine learning to this.
Someone is. It is called generative chemistry It exists, it is genuinely useful in some contexts, and it is partially overhyped in others.
A key concept in SAR is the Bioisostere. When a functional group causes problems, it is metabolically labile and gets broken down too fast. It introduces toxicity, it collapses solubility. You replace it with a Bioisostere, a different chemical group with similar size, shape, and electronic character, but improved behavior in the relevant property.
Like a substitute player?
A substitute that must play the same structural position, interact with the same binding site features, but has a better metabolic profile.
The canonical example is fluorine substitution. You have a position on your molecule where a cytochrome P450 enzyme likes to oxidize. It grabs the carbon-hydrogen bond and hydroxylates it, flagging the molecule for clearance. You swap the hydrogen for fluorine at that exact position. The carbon-fluorine bond is significantly stronger than the carbon-hydrogen bond. The enzyme cannot break it. Your metabolic stability goes from a half-life of 20 minutes to a half-life of 8 hours. One atom
changes whether the drug gets destroyed in your body or not?
One atom is the difference between something you take every 15 minutes and something you take once a day. One atom changes the entire dosing schedule.
One atom change, one new patent. Structurally distinct from the original molecule, independent patent term, extended exclusivity. I am not advocating for this. I am describing a documented practice.
It is mildly provocative that the most commercially significant unit in modern drug development is occasionally a single fluorine atom.
SAR also maps selectivity. Your target protein belongs to a family. An enzyme inhibitor targeting kinase A must demonstrate that it does not equally inhibit kinases B, C, and D, because those off-targets may be responsible for toxicity, and you won't know which inhibition is therapeutic and which is harmful.
Selectivity is honestly harder than potency. Potency you can often chase with additional interactions in the binding site. Selectivity requires understanding exactly which structural features of your target's active site differ from the family members, and exploiting that difference at the atomic level.
What if you cannot make it selective?
Then you explore allosteric sites, regions of the protein that are unique and not conserved across the family. Or you abandon the scaffold entirely and start from a different hit. This decision happens constantly in real programs, and it sets timelines back by years.
I once tried to fix a bad chair by adding more cushions. Eventually, I got a different chair.
That is an accurate description of scaffold hopping, yes. Lead optimization is a simultaneous improvement problem across 5 dimensions: absorption, distribution, metabolism, excretion, and toxicity. ADMET. Improve potency without hurting solubility. Improve metabolic stability without increasing lipophilicity to a level that causes accumulation. Improve selectivity without losing potency. There is no perfect molecule. There are only acceptable trade-offs.
This is the most honest description of drug discovery I have heard in my career. It is also deeply expensive.
About 2.6 billion euros on average per approved drug, when you include the cost of all the failures, and that number has been going up.
I need a moment.
The key in vitro assays during lead optimization include metabolic stability in liver microsomes. You incubate your compound with liver enzymes and measure how quickly it disappears. CACO2 permeability, a cell monolayer model of the gut wall that predicts intestinal absorption. Plasma protein binding, because only the unbound fraction of your drug in plasma is free to reach your target, and hERG channel inhibition.
What's HERG?
The hERG potassium channel regulates cardiac repolarization. Inhibiting it disrupts the electrical cycle of the heart and can cause a fatal arrhythmia called torsade de pointes. Many structurally diverse compounds have hERG liability. It is a hard kill criterion at most pharmaceutical companies. If you inhibit hERG potently, your compound does not proceed.
There is a cardiac death screen?
Performed routinely on every lead compound. Multiple drugs have been withdrawn from the market because hERG liability was not identified early enough. The assay exists precisely to catch this before human studies.
The ERG gate has ended more promising lead series than I can count, including several I was personally attached to. The protein has a very large, hydrophobic binding cavity that promiscuously accommodates lipophilic basic compounds, which is exactly what medicinal chemists tend to make when they are optimizing potency. It is an adversarial relationship.
It is mildly sobering that the expected development cost per drug is so high that it structurally disincentivizes investment in diseases affecting small patient populations, regardless of unmet medical need.
The final milestone in lead optimization is the preclinical candidate. The compound has satisfied pre-agreed criteria across potency, selectivity, ADMET, and safety, in vitro toxicology, in vivo pharmacokinetics, and advances into formal development, toxicology studies, formulation work, regulatory package preparation, and eventually a first-in-human clinical trial.
And what is the success rate from there?
Approximately 90% of drugs entering phase 1 clinical trials fail before market approval. Of those advancing to phase 2, the first efficacy studies in patients, roughly 70% fail. The most common reason is not safety, it is efficacy. The compound does precisely what it was designed to do at the molecular level, and it does not produce the expected clinical effect in the patient.
This is the part that genuinely breaks me. The chemistry worked, the pharmacology worked, the biology just did not translate from the model. Target selection failure, patient stratification failure, biomarker failure. The molecule did its job. The hypothesis was wrong.
It is mildly humbling that the gap between a compound behaving correctly in a microplate and a compound helping a human being is where the majority of investment and the majority of careers in this industry terminate.
Which is why target validation, understanding whether your protein target is genuinely disease-causal in humans, not just disease-associated, is arguably more important than everything we have discussed today. But that is a different episode.
I have done informal target validation in the shed. I won't specify the targets. Legally speaking...
Petar, you have been quiet for a while. What do you think?
You look for something that sticks. You check it isn't lying, then you change small things until it sticks better, lasts longer, and doesn't destroy the heart. Then you check it works in a person. Usually it doesn't, but sometimes it does. That's the drug.
Petar, that is the entire drug discovery pipeline. That is the whole episode in 6 sentences.
That is an accurate summary, including the cardiac safety element, which is the most frequently underestimated bottleneck in the field. I am choosing not to be as surprised as I could be.
The heart part. She just folded the Herg assay into doesn't destroy the heart and moved on. That's 40 years of cardiotoxicology in 4 words.
Usually it doesn't, but sometimes it does. I'm putting that on a slide for my next LP meeting. It is also a perfect description of a venture portfolio.
It is mildly inevitable that Petar's summary is more comprehensible than the preceding many minutes.
Final exam. This episode had real numbers in it, and I am ready.
Pause the video now. 3 questions. Answer before you press play. Question 1, what does pain stand for? And give 2 mechanisms by which pain's compounds generate false positives in a high-throughput screen. Question 2, state all 4 criteria in Lipinski's rule of 5. Question 3, what is a bioisostere? Give 1 example of a bioisosteric substitution used to improve metabolic stability in a drug lead.
Take your time. Think carefully.
We will wait.
Question 1, pains. Pan-assay interference compounds. Any 2 of the following mechanisms, fluorescent interference with assay readout, covalent nonspecific reactivity with assay components or protein, and aggregation-based nonspecific sequestration of the target protein.
I wrote, they cheat on the test, and I stand by the conceptual accuracy of that answer.
It is not technically incorrect. Question 2. Lipinski's rule of 5, molecular weight 500 Daltons or less, logP 5 or less, hydrogen bond donors 5 or fewer, hydrogen bond acceptors 10 or fewer.
I got 3 of 4. I always forget the hydrogen bond donor count, because I find hydrogen bond donors to be less commercially interesting than logP.
That says a lot about you.
Question 3. A bioisostere is a replacement functional group that maintains similar size, shape, and electronic properties to the original while improving a specific physicochemical or metabolic property. Example, substituting fluorine for hydrogen at a site of cytochrome P450 oxidation. The carbon-fluorine bond resists metabolic cleavage, increasing metabolic half-life without substantially changing the molecule's interactions with the target binding site.
I wrote swap an atom so it doesn't get eaten, and I feel that is the grade 6 version of exactly correct.
I submitted a novel Bioisostere I developed in the shed last year. I am confident it deserves partial credit.
I answered question 3 with patent the fluorinated analog and file before the original compound's exclusivity lapses. I maintain that is the applied answer.
It is mildly